So, do folks have any questions from our discussions that started on Friday, it was? Any questions from Friday? Before we get further into today's lecture, I've been asked by Eli and AJ to announce that there will be a review for the fourth exam. It'll be this Wednesday, correct, guys? This Wednesday at 5 here. So if you want to be part of the review session, come to class on Wednesday. It'll just roll right on. Maybe bring a snack so you can have a little snack in between or whatever. And I imagine you guys will probably have handouts for that as well, right? So it should be a good event. Everything I've heard from the previous ones, they were well worthwhile. Any questions about the review session or the exam? I'll be asking Brenda shortly to put on icon last year's final exam. Is the 4th hour exam up yet? Has anybody looked to see it? Is it up? Yes? No? No one's looked. Okay. I will make sure that that's up as well. And I may release the review handouts in advance of those lectures so you have a little more time with them. The ones from last year, they may be modified a little bit this year. Okay. I think actually we talked about this last time, yes? Okay. So we'll go on. Now, when we think about the molecule, the RNA molecule, the messenger RNAs, or actually any RNA, RNA molecules are very rarely used the way they're transcribed. They get modified. And this is certainly true in eukaryotes. It's generally true in prokaryotes in many aspects as well. And some of these modification processes actually happen while the molecule is still being transcribed. That is, the cell doesn't wait for it to be completed and then start processing it. Processing and transcription are kind of an ongoing, simultaneous event. And to show you an example of just how intimately associated they are, some of these processing enzymes actually bind to that C-terminal domain, the negative noodle on the polymerase, so that the tail of the polymerase can have some processing enzymes on it to capture that newly made molecule. The 5' end of eukaryotic messenger RNAs is a pretty peculiar little beast. It has something called a 5' cap. And to modify the original 5' end into the kind that it has to be functional as a messenger RNA in eukaryotes, 3 different things have to happen. So it's going to trim off the pyrophosphate tail. It's going to add on a so it's going to trim off part of the phosphate tail. It's going to add on a guanine backwards. That is, instead of adding it on so it's on the 5' end, it now has a free 3' end where it used to have a free 5' end. And then the guanine itself has a methyl group added to it. So this is often called the methyl G cap. And what it means is that a messenger RNA in eukaryotes has 2 3' ends as far as the chemistry of it's concerned. And so that's one of the ways in which the cell marks it as being available for translation and helps to stabilize it, that the 5' cap both modifies its effectiveness and its stability. The other end is also modified. Obviously can't be modified until after transcription. The 3' end gets something called polyadenylation. And so there's a cleavage enzyme that can cleave after a sequence in the RNA transcript AAU, AAA. And then this enzyme can add additional A's on that cut end. And the adding enzyme is just making a string of A's, so it doesn't need a template. It adds just to the end of a free-floating molecule, no template needed. The chemistry of the reaction is the same, but there's no template needed. And when the messenger RNA has that poly-A tail, there is a protein called poly-A binding protein. You could have guessed that if it's been added, there's going to be a protein that will recognize it. So the poly-A binding protein recognizes this poly-A tail. It can be anywhere from 20, 50. I've seen poly-A tails as much as 100 or 200. And when the poly-A binding protein is bound to the mRNA, that is one of the things that also contributes to a longer lifetime for messenger RNA. It also, interestingly, improves the translation of the message, which, if you think about it, is a little odd because it's at the other end. And we're going to see how the 3' end could modify efficiency of translation. There are a few messenger RNAs that do not have poly A, so their structure is such that it does not attract the enzyme to add the A tail. They tend to be very short-lived, and by not being polyadenylated is one of the regulations used to have them be only around for a little while. And so it's one way to tune them up fast and down fast. There's another reminder again. I know some of you probably weren't here Friday, so this is just a reminder that we will be covering all the chapters on the exam on Monday, May 4th. And the last 2 sessions will be used for review and integration. And the final exam will be Monday, May 11th at 2:15. Now, we talked earlier about several common DNA binding proteins that are involved in regulation of transcription. But there are all other ones that are could almost be considered sort of architectural proteins. And these are some examples of those guys. So here's the TATA box binding protein. Here's the integration host factor, meaning it's involved with whether a virus can integrate. Here's something called HMG, which stands for high mobility group. It's a bigger protein that's involved in the chromosome. And SRY rather, is involved in sex determination. And all of these now bind at the minor groove. You remember before, the things that seem to be transcription factors, they bound at the major groove where there were exposed hydrogen bond acceptors and donors. These more architectural ones that actually can change the shape of the DNA bind at the minor groove. So a different kind of a structural strategy. So we've talked in talking about RNA synthesis, we've talked about initiation and how cells decide where to start and how much to do. We've talked a little bit about elongation and the fact that the CTD needs to be phosphorylated. It's a very processive process. But you have to end it also. DNA replication has initiation and elongation. It just goes around to completion. RNA synthesis, you don't want the whole thing done. You've got to stop. And just simply stopping is usually not sufficient. A pause is not usually enough to end the transcription. If we look in prokaryotes, about half of the genes have things that are called terminator sequences. And these are sequences that have the sequence structure that permit the formation of a hairpin. That is, that as the RNA is synthesized, that it comes up into make a double strand within the molecule and then a little bit of U following after it. And that seems to be one of the signals in prokaryotes. In eukaryotes, things are a little bit still up in the air. That C-terminal domain that I mentioned before, this is a different C-terminal domain, excuse me. The polyadenylation factor that can polyadenylate transcripts has a domain in it at its C-terminus that seems to contribute to polymerase II termination and does not play any role in the RNA processing part. Some other proteins that have been sort of had a question mark added to their name as perhaps playing a role in RNA synthesis termination are these cell cycle associated proteins and another protein that plays a role in chromatin structure. In Saccharomyces cerevisiae, this is a yeast that makes a model for a lot of general principles of how eukaryotes work. In it, many of the large genes have a physical association between the promoter, which is supposed to be the signal to start it, and a terminator region where you want to finish it, so that the various kinds of looping architecture that we've talked about can actually organize the gene structurally to make sure that the complete domain is all in one loop by itself. So there's suggestions there of things that we may yet learn. In eukaryotes, most eukaryotic genes have something called an intron. An intron is, jargon for intervening sequence. And it's an intervening sequence because it's not going to be there in the mature, final, translated messenger RNA. Now, how many introns a gene has will vary with the complexity of the organism, and this is one of the things, for example, that can contribute to genome size, because the more introns you have, the more stuff you need that's not going to make actual protein. And here's an example showing you how that might work. Here's yeast that almost all their genes have an intron, but not more than one. Here are fruit flies. They average about 2. But they've got some that have, 7 or 8. They've got a couple that have 30, 40, or whatever. If we look at mammals, the average is quite a bit higher. There's sort of several different peaks. There's one peak here around 5, another one around 9. And then there are examples out there with 30, 40, 50. The titin, the protein that makes muscles organized as nice sliding things, has a molecular weight of the final protein of 1 million molecular weight. You can imagine how many introns its messenger RNA must have. And it turns out, actually, to the point where in mammals there's more intron than exon in many genes. There it is. And if we look at the structure here, you don't need to turn quite yet. Here's an example of a typical intron within the context of the messenger RNA before it's been processed. And it's just sort of flagging your attention to the fact that there are sequences that are signals for where the intron begins and ends. And so there's sequences at the 5' end of the first expressed domain that straddles the splice site. There are sequences at the end of the intron that straddle the splice site, and an A in the middle that's cryptically called the branch point. And we're going to see what that is. But it's about 20 to 50 bases from the end of the intron that's going to get spliced out. And so what has to happen for these introns to be cut out of the RNA and to make a new continuous molecule of RNA is we're going to have to splice out that little piece of intron, or sometimes big piece of intron sequence. So now remember, we're looking at the RNA molecule. This is the transcribed RNA molecule. And there's going to be at least a 2-step process where the 2' hydroxyl, so in RNA like in DNA, the 3' and the 5' hydroxyls are busy, but the 2' hydroxyl is free. So the 2' hydroxyl can attack here at the terminal G of the initial G, I guess you could call it, of the intron, and end up making a looped molecule. You see here how what was a phosphodiester bond to that part of the expressed sequence is now a phosphodiester bond to the 2' hydroxyl, leaving a free 3' end here. This free 3' end can now come in and pick up the other end of what we want to make as an intact, sealed together, fully spliced RNA molecule. And so when this 3' hydroxyl comes in, you now have an intact RNA molecule, and you have this intron bit that is detached from the original molecule and has a molecular loop in it. This is sometimes called the lariat because it looks kind of like the lariat that cowboys would use to get horses or calves and stuff. And since these bonds are you're changing one phosphodiester bond into another phosphodiester bond energetically, it looks like it ought to be fairly equivalent. Sometimes it will be using some energy, but that's a variable thing. This ability of messenger RNAs to be spliced, to have introns in them that then get taken out, turns out to be a very important part of how mammals, including us, can make a lot of variety out of a smaller number of genes. Before we sequenced the human genome, we thought we had 100,000 genes. Looks like we're down to about 20 or 25,000 genes, but those genes have a lot of introns in them, and by selectively splicing it in different ways, in different tissues, we can make a lot of different proteins. And this is just sort of an example for you. This particular one I think is taken for tropomyosin. The exact protein doesn't matter too much. But what's relevant here is you're looking at several different tissues. So here is the gene as it exists in the genome, replicated every time a cell divides and so on. And they've color-coded some of the expressed sequences as different things. And then the white boxes are going to be introns in some muscle, in some molecules. So when you transcribe this gene, it gets transcribed clear through, and all that stuff is in it. In striated muscle, splicing happens in such a way that it selects the lavender and not the red box, and it does include the orange box, and it includes both of these lavender boxes. So striated muscle would look like that in the mature spliced message. In smooth muscle, it picks the red box and doesn't include the lavender box, and it doesn't include these two extra bits there. So the protein it has this bit down there. So the smooth muscle message is different than the striated muscle message because it's been spliced differently. And non-muscle fibroblasts have a shorter final message still. Here's brain. Brain and non-muscle fibroblasts have the same protein sequences, but the non-muscle fibroblasts have a longer untranslated region. So that can also affect how a cell uses a messenger RNA. So differential splicing, where one transcript gets a different processing in different tissue types, very important part of how the human body can have as much variety in its proteins. Now, there are several different kinds of introns, and they get spliced by different mechanisms depending on what they are and where they are. So type 1 introns are self-splicing. That is, these are things that can handle their own rearrangement to become a final process thing. We see introns in a number of places in eukaryotes, nuclear, mitochondrial, chloroplast genes, so all three organelles, if you like, of eukaryotes. And the kinds of things that have them include, again, a variety of RNAs, ribosomal RNAs, messenger RNAs, tRNAs. Interestingly, type 1 introns also are found in archaea. And you may remember archaea as that group of prokaryotes who are kind of different from some of the other prokaryotes and have odd chemistries and so on. They may, in fact, have a relationship by descent to the eukaryotic cells. Type 2 introns are only found in the mitochondria and chloroplasts. So fungi, algae, plants, archaea, things like that. Type 3 introns, these are only found in the eukaryotes. Type 3 introns need machinery to help them do it. They use small nuclear RNA proteins, or SNRPs, small nuclear ribonucleoproteins. And the SNRPs get assembled into something that has been called a spliceosome, a little body that's involved in making the splicing work. A fourth kind of introns is found only in transfer RNAs. And this kind of splicing needs energy input and an endonuclease. This is also found in the archaea. So we have a couple of places where archaea share a molecular mechanism with us. So the reason you don't see a lot of stuff here about bacteria is bacterial cells don't have a lot of introns. The archaea have some, but apart from that, the prokaryotes make the RNA that they want. This is showing you schematically how some of those introns work. You may remember that group 1 and group 2 were both the self-splicing kind. And we talked before about, excuse me, I should not cough into my hand during a swine flu epidemic. That's a little better. The right way to cough, if you need to do this class. A lot of people died the last time we had this flu, so let's be good. Okay? It's not in Iowa yet, but we can all be careful anyway. You didn't know? There are people here who didn't know yet? There is a swine flu epidemic that's starting up. Mexico has had a number of deaths. What's of great concern is the Mexican deaths are in young people, 20 to 30, which is usually the hallmark of a flu that is so new to the population that it induces something called a cytokine storm. That is, instead of just kind of the ordinary, let's get rid of this virus immune response, the body mounts every bit of ammunition it has, and you're attacking yourself rather than a virus. So keep an eye out for it. If they have public health measures, do what they tell you. I think we can handle it, but it's not a good thing. Here's the group 1 again, to get to something cheerful. So we have a transesterification where we tack in, and a second one cleaves that part off. So in the case of the type 1 group, it may be self-splicing, but it actually doesn't make a lariat. It's 2 transesterifications, but it doesn't make a lariat along the way. The type 2 was the one that we saw in the first diagram, where the A attacks to make a branched point there, and the new 3' hydroxyl attacks to re-anneal the messenger, the RNA molecule, and you have now a free lariat rather than just a scrap of RNA. In the type 3 group, the chemistry looks similar to the type 2, but as I mentioned, this is going to need the assistance of a lot of other proteins. You know, if you think about our cells with their meter of DNA and all that stuff going on, you probably don't want to let it just kind of bumble around and see if it finds the right place to splice. Part of the job of the spliceosome is to handle the correct splicing as opposed to sort of some splicing. This is showing you the tRNA splicing I talked about. So it cleaves here. So it has kind of extra RNA there. It cleaves there. And the kinase and ligase can re-anneal it at the expense of ATP. This is a figure that shows you in a little more detail what those type 3 splicing events look like. Here we have a template of the DNA. Here we have an RNA polymerase II. So this is the RNA polymerase that makes messenger RNAs. And here's a growing RNA molecule, an exon here, an expressed sequence that will be in the final messenger RNA. And here's an intron that's being released from the polymerase. And here moves in one of these SNRPs with U1 RNA on it. And the U1 SNRP can bind at the first junction. So you see here the 5' splice site. The polymerase continues to make a longer and longer molecule. U2 comes in and binds near the junction. The 3' splice site is emerging, has just recently emerged from the RNA. And you have U4, U6, and U5 coming in, handling the rest of the work and coming together to make everything go forward. This is a diagram that tries to show you how, remember that SNRPs, small ribonucleoproteins, it's both RNA and protein as a particle. This is just showing you how some of the RNAs in the SNRP interact with the messenger RNA. Question? Okay. And you can see here how you have the, here's the exon 1 here. This is intron here. There's exon 2. So this is your messenger RNA here. And it's interacting with the U6 RNA and the U5 RNA and the U2 RNA. So the RNA molecules or the SNRPs are helping to hold that complicated RNA in the right configuration for this to go forward. And since some of these introns can be longer than the expressed sequences, you need to really have machinery to keep track of it all if you don't want it to end up like a bowl of spaghetti. Now, as I've said before, degradation is always as important as synthesis. And nothing's just as true for RNA as anything else, although it took us a while to realize that. Here we have a diagram on the top of a mature messenger RNA molecule. It has its 5' methyl G cap at the 5' end of the message to make it kind of look like a 3' end. Here it's got its poly A tail of 20 to 50, 100 extra A molecules at the 3' end. And the time has come for this thing to die. Typically, the degradation will start at the poly A tail end, and an enzyme called deadenylase can simply eat in until it eats off the poly A tail. When the message has lost its poly A tail, it's now vulnerable to another enzyme called a decapping enzyme. And this may be why messenger RNAs that don't have poly A to begin with are short-lived. The decapping enzyme can cut off that methyl G cap, and now it has an exposed 5' end. Remember, the methyl G cap kind of looked like a 3' end. Now it has an exposed 5' end, and at this point, the molecule can be attacked from both ends. There's a 5' exonuclease that eats free 5' ends of RNA. There's a 3' exonuclease that eats free 3' ends that aren't all A, and they just kind of eat in until the message is gone. There's another dimension of controlling RNA stability that's of fairly recent discovery, and we're really still in the process of fully understanding its significance, but it's clear that it's going to be important. And it's something called RNA interference. And the shorthand for it is something called small interfering RNA. And they're called that because these are small molecules of RNA, and they interfere with the stability of an RNA molecule. And these small interfering RNAs turn up in several contexts. They were discovered in some that seemed to be kind of specialized initially. For example, there's something called nonsense-mediated decay. New messenger RNAs are, in effect, proofread. They're kind of examined for overall quality of fit. And if there are nonsense mutation in there, so they can't be properly translated, they'll be degraded. And small interfering RNAs play a role in nonsense-mediated decay. A second place where we started finding out about small interfering RNAs, or siRNA, was in the defenses that the cell has against double-stranded RNA viruses. Viruses come with all kinds of nucleic acids. There's some double-stranded DNA, some single-stranded DNA, double-stranded RNA, single-stranded RNA. Double-stranded RNA viruses, small interfering RNAs, are one of the tools that the cell has to defend against that. siRNAs were found in these contexts, but as we started to have better tools for identifying them and for studying them, it's turning out that they also probably have a role in development, and that role in development will probably continue to grow. So if you have an interest in that area, I would keep your ears perked up for that. And this is one sort of generic representation of how a small interfering RNA works. Here we have a double-stranded RNA that's going to be producing a small interfering RNA. An enzyme called dicer cuts it up, you can see, kind of like you would with a little dicing chopper. If siRNAs have an appropriate mRNA target, the complex of those together will attract or activate an RNase that's specific for that complex of them, and therefore degrade the messenger RNA for which that small interfering RNA is the ligand. This is showing you this in a slightly more detail. You can use this as a tool. That is, you can create a small interfering RNA intentionally or by design. And so let's say we've just made a synthetic small interfering RNA, a little 20-25 nucleotide one. If it combines with the other proteins of the complex to form the full complex, it can then recognize the single-stranded RNA to which one partner of that is complementary, and if it then binds to that single-stranded RNA in that effective complex, the target will be degraded, and interestingly, the small interfering RNA is not used up in the process. And so one of the things that makes small interfering RNA powerful is that it can be used, in effect, catalytically. One molecule of small interfering RNA can degrade many molecules of messenger RNA. And how good is it? Well, we can use this in tissue culture, and we can use this in animals to create knockouts where we turn down the expression of something. And this gives you sort of a visual representation of how good that can be. In this cell on the left-hand side, it's a fluorescence micrograph, and you can see the green color is the protein of interest. And here we've incorporated some small interfering RNAs, and it's pretty dang low. think even with the overheads, you can see that that's pretty dang low. There are some who hope that perhaps we can use this even for gene therapies. Now, whether we'll get that far, I don't know. But you can see how it would be attractive to have something that's this specific, because it will only interfere with the thing for which it has the complementary sequence. This is then showing you the machinery of it in a slightly larger blow-up of it. The small double-stranded RNA that's the complementary to your ultimate target is going to have to be unwound to make to hybridize to its complement that it's going to be attacking. RISC stands for RNA-induced silencing complex, and the RISC can act in a couple of different ways. If the two strands are really good and complementary, the RISC complex can cleave the RNA and lead to its degradation. If the two strands, this siRNA and its target, are not perfectly matched, so like a bubble sort of thing, the RISC complex simply stays bound to it, doesn't cut it, but also doesn't come off, and it can't be translated. So in both cases, you're going to end up preventing the expression of a protein from that RNA molecule. And this is sort of, I love the way medical illustrators do these things. So here the DNA has been transcribed to make a double-stranded RNA, the microRNA that's going to be producing the siRNA. And here's your dicer enzyme that's using lightning bolts to chop off pieces of small interfering RNA. So you got to love them. This is showing you how you could do it by doing it artificially, that you could simply dump into the cell small pieces of RNA that would be the right size compared to what dicer would make for you. We can show the potential for this to be clinically interesting by trying experiments in mice. You may remember LDL. LDL was the bad kind of lipoprotein when we were talking about cholesterol back in the lipid chapter. There are genetic syndromes in which very, very heavy elevations of LDL are associated with very early and bad heart disease. And the idea was, can we use small interfering RNAs to turn off ApoB protein genes? These are the genes that code the protein component of the LDL. And one hope here was to, for instance, maybe help get the small interfering RNAs in by coupling them with cholesterol as a way to get them in. The good news is that, in fact, you can reduce cholesterol ApoB1 expression. You can reduce total cholesterol. So these are all good things. The bad news, it only worked in one mouse out of 84. So development is needed. That's why I say it shows potential rather than promise. So far we've been talking about the processing of messenger RNAs, which, because messenger RNAs are so diverse and because we do have tissue-specific processing of them, present some of the biggest challenges. But the other RNAs are also processed. Both ribosomal RNA and tRNA are not functional until they've been modified. Now, ribosomal RNA, you may remember I said, is the most abundant RNA in the cell, about 80% by mass or more. It even has its own special structure in the nucleus where it's made. So the nucleolus or nucleolus, depending on who you learned it from, is where ribosomal RNA is synthesized, and it has its own polymerase. Polymerase 1 is the one that synthesizes ribosomal RNA. And it's going to have to be, it has introns in it, and it's going to have to be cleaved and trimmed. If we have, for instance, here's a 45S ribosomal precursor molecule. There's no molecule this big in ribosomes, and so what's going to happen along the way is it's going to have to be cleaved. So this red piece will make it into the ribosome, the yellow one will, and the green, but the rest of it's going to go. And many of the bases are going to be chemically modified. These are some examples of the kind of chemical modifications. Here's pseudouridine. where you've, after you've, let me think. I'm forgetting the exact chemistry of making pseudouridine, but it is one of the modified bases in ribosomal RNAs. Methylation of other bases can happen. You don't want the modifications to be random. So SNO RNAs, or small nucleolar RNAs, are involved with guiding the correct site on which those modifications can happen. The 3 ribosomal RNAs are not by themselves sufficient to make a functional ribosome. There's about 80 ribosomal proteins that are going to have to associate with those RNAs correctly. So the nucleolus is this factory for producing the subunits of ribosomes that involves both the processing of the ribosomal RNAs and the assembly of them with the subunit proteins. Transfer RNAs have their own polymerase. So transfer RNAs are made by polymerase 3. They're also pretty abundant, more abundant in mass than messenger RNAs. They also need to be cleaved and trimmed. And interestingly, the enzyme that performs that is something called RNase P. And RNase P is interesting because the catalytic site, the actual active residues in RNase P, are RNA. This is one of the examples of a catalytic RNA in eukaryotic cells. Some people think this might be a residue of the old RNA world, if that's where things got started. tRNAs are very heavily modified. A quarter of the bases are modified. And these are, I mean, this is quite a zoo of modifications. I don't expect you to remember all of them, but this is sort of an example of the kinds of changes that can happen. You can have methylation happening. You can add isopentenyl chains to them. Here's dihydrouridine. It's been reduced. Here's 2 methyl groups have been added here, so quite a lot of modifications can happen. Some of the tRNAs, but not all, are spliced. Some of the ones that are spliced can be self-spliced. And in eukaryotes, there's a CCA that's added at the tail of every tRNA. Just as we add lots of A's at the end of most messenger RNAs, there's a CCA added at the end of every tRNA. I'd like to sort of introduce you to the idea that RNAs actually have this possibility of being enzymes. And I want to just see where we are in terms of time and future and stuff. We've already met one catalytic RNA in RNase P. We're going to see more when we get into the ribosome itself. And the chemistry of RNA as a molecule can provoke these thoughts of noticing similarities and stuff. So there are several things that RNA does that proteins do, and in proteins, these are the things that make proteins good enzymes. So they both have a flexible backbone. RNA is typically present as a single-stranded molecule and has the flexibility of a peptide backbone. RNAs can, self-associate through non-standard pairing, just as proteins can self-associate through hydrophobic bonding. RNA molecules and protein molecules both have tertiary structure. They're finite length molecules with an exact sequence, and when they finish doing all their folding and stuff, they have a particular tertiary structure, maybe 2 or 3 tertiary structures. They don't have an infinite zoo of tertiary structures. They're both capable, some of them are capable, of metal binding, of having rearranged these side things sticking out in ways that can bind a metal ion. So these structural properties imply that RNA has the potential to be a catalyst. That was first discovered with these self-splicing RNA molecules. And for a while we thought that self-splicing RNAs were the only kind of RNA catalyst there was. These diagrams, by the way, are showing you the non-standard base pairing. These are what are called non-Watson-Crick base pairs, where you can see here A and G actually base pair, or here you have a threesome with AA and UAA and so forth. So these non-standard base pairings also contribute to the tertiary structure of things like tRNA and ribosomal RNA. I stand corrected, but the very first ribozyme that was called that was a plant virus, an RNA virus that was processed into the final size in the absence of proteins in the test tube. I'm putting a little caveat in here calling it self-cleaving, and I don't like calling it a catalyst because it was used up in the reaction. That is, the first thing that was called a ribozyme wasn't an enzyme because it was consumed, but it did do the reaction by itself. We do now know authentic RNA catalysts, RNA molecules that can accelerate a reaction multiple times and not get used up in it. This is showing you the structure of what's called the hammerhead ribozyme, that first self-cleaving RNA that was found, and how the tertiary structure of the initial molecule created the positioning of the parts of the molecule in a way that the cleavage reaction could occur. So that actually finishes the material that was in your textbook, but I want to see if I can share a little bit of what's called the RNA world with you. And if we don't get to the end of the handout at this point, don't fret. I won't expect you to make sense of something I didn't get a chance to talk about. The phrase the RNA world is used to describe the idea that there may have been a biochemistry of life earlier than ours and simpler than ours, in which RNA was a much larger part of what went on. And the reason for suggesting this was based on reflections on what our current life is like and how hard it is to imagine it to evolve. If you think of life as it exists right now, it's very busy. The central dogma says information in DNA can make more DNA, information in DNA can make RNA, information in RNA can make protein. The red arrows are simple. What's messy is that we need the other components for most of these things. So for DNA to make DNA, you need RNA and protein. For DNA to make RNA, you need RNA and protein, and so on. So you're using the components of the system to make the system. It's what can be called reflexive or recursive. It really seemed like a problem to people how you could ever get something like that started. If you need the product to make the previous product, how do you ever get it started? And in fact, I was baffled myself when I was your age. To make life worse, this whole thing has to happen inside of something. You need an inside and an outside. You need some control over what you're doing stuff with and so on. So on top of this complicated recursive system, you have to account somehow or other for a selective boundary. And so thinking about the notion that you might be able to start with something simpler, several people a number of decades ago said, well, maybe if we started with a system that just uses RNA for all that stuff, because RNA can carry information, and maybe RNA could be a catalyst, you know? And that then gave rise to other questions. So, for example, how could you have replication without recursion? Now, replication means copying information. Recursion means how can you get that to happen without having already done it? Is there any way to get the thing started? Is there any way to do it without using the thing you're making? Some specific, just kind of practical questions about an RNA world were, how can you get the RNA to last long enough to do anything? If any of you have worked with RNA in the lab, you know you've got to be very nice to your RNA. It tends to go away if you're not nice to it. And one of the difficulties I always felt thinking about the RNA world was, how would it ever last long enough to do something? There are hints and suggestions. Another is, what reactions could RNA catalyze? I mean, is this going to be a very limited, boring world where RNA molecules just splice RNA molecules, or is there something more general going on? And finally, how could you have self-regulation? Right now, the RNA depends on the DNA and the protein to control how much of it there is and so on. Could it conceivably be self-regulating? And then, if you can put together a simple system using RNA for all of these functions, how would you get a boundary around it? Well, there was a fascinating and revolutionary paper in 2004 on something that we can call molecular midwives. And this is something that Karen Smith had predicted probably 20 years earlier. And his comment on the apparent impossibility of getting life started he made an analogy. If you think about an archway, have you ever seen these stone archways with a keystone in there and this lovely stone arch? And you think, how could that thing have ever gotten there? Because, it needs everything for it all to be there. Well, of course, when they built it, they had a scaffolding under it. And they put in the keystone, and once they put the keystone in, they could take the scaffolding away. These molecular midwives may be the scaffolding that permits this kind of a supporting structure to happen. This molecule, called proflavin, is able to stabilize two RNA molecules or two DNA molecules next to each other to facilitate replication until the bond forms. So this is a kind of replication that doesn't depend on the presence of enzymes. Proflavin looks chemically like this. It's obviously an aromatic hydrocarbon, an aromatic heterocycle for that matter. Proflavin occurs abiogenically. You don't need living systems for it to occur. It's found in the environment. It's found in meteorites. It kind of has the same shape as a Watson-Crick base pair. So here's the drawing. Here's the base pair. You look at the space-filling diagrams of these two things, and you see that the two have a remarkably similar footprint, especially that they both have this aromatic character for stacking. And so what can happen is this. If you have an initial template strand and you have substrates that you want to hybridize to that, in the presence of proflavin, it can stack between those bases and facilitate those interactions until the bond forms spontaneously. This can accelerate the polymerization reaction by 1,000-fold. So not as good as DNA polymerase, a lot better than waiting around for it to happen by itself. This is just a curve showing you that as you increase the amount of proflavin, you can get more product made. It's always nice when you can show concentration dependence in experiments, and it does. So this is the polymerized product here. And most of the product is the right molecular weight. That's good. This is showing you the rate of reaction as you have a change in concentration with the intercalator, and it's effective at really quite low concentrations. These are micromolar concentrations, so you don't need very much of it. In fact, more of it's worse. Just need a little bit of it around. And you can get, as it says, in the order of 1,000-fold accelerations. I think given the time, I'm just going to skip over this. If people want to talk about it, come to me afterwards. The chemist doing up the math on it suggested that there were 3 proflavins per complex in the way this experiment was set up, or that 1 proflavin sat between every 2 bases to facilitate the binding. So a very elegant thing. So we found a possible solution to how you get replication without depending on the product of replication. This is a molecular midwife who solves that problem. And this, I think, is just simply repeating what I've said so far. So now we possibly have replication. We still have this question about can RNA ever be stable enough? Can it be interesting enough? And can it be smart enough to provide the basis for a living system? So it needs to be stable. It needs to be flexible. It needs to be smart. And it turns out that a very simple molecule out in the environment radically improves the stability of RNA. There's a kind of salt called borate. If you've ever used borax, you've used it. And if you don't have borate around and you start trying to synthesize sugars randomly, you can take small carbon fragments like glycoaldehyde and get things to start to smush together. But once you get above 3 or 4 carbons, you start to get brown goo. What the interesting thing about borate is that borate favors the formation of sugars compared to brown goo. Borate can stabilize some of these what are called vicinal glycols. They're high alcohol groups side by side. And because the borate can stabilize that while reactions are happening, you can add another little carbon piece in, and you can end up with extremely good production of 5 and 6 carbon sugars if there's borate mineral around to protect the growing sugar, prevent the cross reactions, and so on. This is just showing you some of the sugars that were produced in the presence of borate. This is some of the cyclic forms. I think we won't go. Okay, it's 20 after. I'll stop. Anybody wants to hear more, please ask. We can cut the recording.